when we think about a cancer gene, there is this massive underlying assumption we all tend to make.
Oh, absolutely.
We view it as this universal bad guy. Like, the logic goes, if you have a specific genetic mutation, it just has a specific, predictable, destructive job in your body, regardless of where it happens.
Yeah, and it is a highly deterministic way to view biology, right?
Yeah.
They've spent decades looking at it as a simple equation. Gene X breaks, therefore it results in cancer Y.
Right. But imagine if the physical location of that mutation completely tore up its job description. What if, say, a genetic typo that causes utter chaos in your breast tissue is just, sitting quietly as a harmless bystander if it happens in your lung?
Well, if that's true, and the data says it is, it forces us to rewrite the entire rule book for how we understand, diagnose, and ultimately treat the disease. Because we can't just hunt for a mutated gene anymore. We have to understand the neighborhood it lives in.
And that is our mission for today's deep dive. We are digging into this landmark 2026 study published in Cancer Cell by Ben Lamudi and colleagues.
Such an incredible paper.
It really is. The goal for us today is to understand how the context of a tumor, meaning the specific tissue it grows in, the evolutionary timing of the mutation, and even your own genetic ancestry, how all of that radically alters the behavior of these so-called cancer drivers.
Yeah, and to even begin this conversation, we really have to grasp the sheer gravity of the data these researchers were working with.
Please lay it out for us.
So this is the MSK-50K cohort. They analyzed 54,331 individual tumors from over 48,000 patients. And this wasn't limited to just one type of disease, It spanned 448 different cancer subtypes.
Just visualizing over 54,000 sequenced tumors is staggering. That is a mountain of genetic data.
It is. And they used a clinical sequencing tool called MSK-50K.
Right, I've heard of that.
Yeah, it specifically hunts for alterations across up to 505 cancer-associated genes. But honestly, the scale isn't even the most impressive part here.
What is?
It's the methodology. They didn't just sequence the tumor tissue. They took the tumor DNA and compared it directly against the patient's matched normal blood DNA.
Okay.
Yeah.
I want to focus on that matched blood DNA for a second because I can guess why that's so crucial for the listener to understand.
Go for it.
Every human body is full of harmless, inherited genetic quirks, right? Like, if you only look at the tumor, you're going to see thousands of mutations that have absolutely nothing to do with cancer.
Exactly.
They're just the normal typos that make you you. So by sequencing the normal blood alongside the tumor, you're essentially creating a filter to subtract all that background noise.
You've nailed the exact mechanism. By subtracting that inherited baseline, the researchers isolate the true somatic mutations.
Somatic meaning?
Somatic just means the genetic errors that were newly acquired by the cell over a person's lifetime, not inherited. Those are the actual drivers we care about.
So if previous cancer databases were like looking at a basic two-dimensional city map where you just see the outline of the streets.
Yeah, exactly.
Taking 54,000 matched tumor normal samples upgrades us to a high-definition satellite view.
Yeah.
Like, we aren't just seeing the streets. We're seeing the cars, the traffic lights, and, who's driving.
That's a perfect way to put it. And the moment they turned on that high-definition satellite view, the very first thing they noticed was this hidden layer of rare anomalies we call hotspots.
Which is a great place to start our analysis. So a hotspot, fundamentally, is a specific recurring site on a gene that is known to mutate and drive cancer.
Yes.
If a gene is a long string of decorative lights, a hotspot is that one specific bulb that keeps short-circuiting across thousands of different houses.
Right. And because smaller, older studies didn't have the resolution of 54,000 matched samples, they only saw the most obvious, brightest short circuits.
The common ones.
Exactly. This massive cohort revealed 164 new, previously invisible cancer hotspots.
Okay, but anytime we talk about discovering new mutations, I have to be a bit skeptical. With a data set this massive, how do we know these 164 new hotspots aren't just random, harmless passenger mutations? Like, how did they prove these specific typos actually drive the cancer?
That's the million-dollar question. They looked at the underlying biology of where these typos were happening. 70% of these new hotspots were located in tumor suppressor genes, or TSGs.
Okay, tumor suppressor genes.
Right. Now, humans usually inherit 2 copies of every gene, one from each parent.
Sure.
For a tumor suppressor gene, those 2 copies act like a dual brake system on cell growth. So to prove these new mutations were actually disabling the brakes, the researchers looked for a phenomenon called loss of heterozygosity, or LOH.
Loss of heterozygosity. Okay, let's break that down for the listener.
Yeah, let's do it.
Hetero meaning different, referring to the two different copies of the gene we get from our parents.
Right.
So, loss of heterozygosity means the cell physically lost its healthy backup copy of the gene, leaving it entirely dependent on the single mutated hotspot version.
Precisely. It is the genetic equivalent of a smoking gun. When the researchers saw these rare new mutations paired with the deletion of the healthy backup copy, they knew it wasn't a coincidence.
The cell was intentionally relying on a broken brake pedal to grow out of control.
Exactly. It's actively throwing away the good brake.
Wow. Can we anchor this in some real biology? Like, what kind of genes are we talking about here?
So they found these newly identified hotspots in genes like KEAP1 and STK11, which predominantly show up in lung cancers.
What do those do normally?
Well, KEAP1 normally helps cells manage oxidative stress, while STK11 is basically a master regulator of cellular metabolism.
Got it.
They also found hotspots in TCF7L2, which is a gene deeply involved in the WNT signaling pathway. That's a major driver in gastrointestinal cancers.
Yeah.
Now, the catch here is that each individual new hotspot is incredibly rare. We are talking about finding them in less than 0.1% of patients.
Which sounds completely insignificant until you look at the aggregate.
Mm-hmm.
It reminds me of the long tail theory in economics, you know?
Oh, how so?
Like a massive bookstore might make a predictable chunk of money on the new Stephen King blockbuster, but they actually generate a huge percentage of their total revenue by selling single copies of thousands of obscure niche titles.
Oh, that's brilliant. Yes.
Cancer is utilizing a long tail of rare mutations. No single rare hotspot is common, but collectively, 5% of all patients in this 50,000-person cohort had one of these newly discovered drivers.
It really proves that cancer doesn't just rely on a few blockbuster mutations. It has an incredibly deep bench of rare, subtle strategies to bypass our cellular defenses.
So just to give a quick recap for everyone listening, bullet one. This high-definition view of 54,000 tumors didn't just confirm what we knew. It revealed 164 rare covert hotspots, mostly in tumor suppressor genes.
Right.
And bullet two. By observing the cells actively discarding their healthy backup genes through loss of heterozygosity, we know these rare mutations are incredibly dangerous.
Exactly.
So I guess the question for you listening is, what stands out to you so far? Is it the sheer number of rare mutations that can drive cancer? Because knowing what these mutations are only leads us to a much stranger question, which is, are they showing up where we expect them to?
And this is where the study gets truly disruptive. It introduces the concept of canonical versus non-canonical drivers.
Okay, canonical. In biology, that basically means the expected traditional rule, right?
Yeah.
Like if a mutation is highly enriched in a specific tissue, it's canonical. PIK3CA is a famous driver mutation, and it is canonical in breast, uterine, and bladder cancers. We expect to find it there.
Precisely. And non-canonical means the mutation is, well, crashing a party it traditionally isn't invited to.
Ah, right.
And the data revealed that a full 1/3, 32% of all driver alterations arise in these non-canonical unexpected contexts.
32%. That is a massive blind spot.
It's huge.
So we are finding that breast cancer driver, PIK3CA, showing up non-canonically in lung and esophageal cancer.
Exactly. And we see another famous gene, ERBB2, which is canonical in breast and gastric cancers, suddenly popping up in melanoma or pancreatic cancer.
But the tissue context is entirely different.
Totally different. The metabolic pressures, the cellular environment, the neighboring cells, I mean, none of it is what the mutation evolved to exploit.
It's like taking a star NFL quarterback and suddenly forcing him to play defense as a linebacker.
That's a great analogy.
He's still an elite athlete, meaning the gene is still a powerful driver mutation.
Yeah.
But the environment, the pressures, and the fundamental rules of his new position are totally different.
Exactly right.
Which leads to the obvious deduction. A quarterback playing linebacker isn't going to play the same game. If a mutation is operating out of position in a non-canonical tissue, it can't possibly behave the same way, right?
It doesn't behave the same way at all. And the fundamental difference in behavior comes down to evolutionary timing.
Timing?
Right. In genetics, we track timing through a concept called clonality.
Clonality. Okay, so that's essentially reading the rings of a tree to see how the cancer grew, right?
Basically, yeah. So If a mutation is clonal, it means it's found in the trunk of the evolutionary tree. It is present in every single cell of the tumor.
Because it happened on day one and initiated the cancer.
Exactly. But if a mutation is subclonal, it's out on a branch. It's only found in a fraction of the cells, meaning it emerged much later as the tumor was evolving or adapting.
Okay, so how does that map to our canonical and non-canonical drivers?
When the researchers looked at the clonality of these drivers, they found a stark divide. Canonical drivers, the expected ones, are usually clonal. They are the early foundational sparks of the disease.
Okay.
But non-canonical drivers, the unexpected ones, are overwhelmingly subclonal. They are late arrivals.
Wait, I want to push back on this to make sure the logic holds. Are you saying a PIK3CA mutation in breast cancer is the architect who designs and builds the house from the ground up?
Yes.
But that exact same PIK3CA mutation in lung cancer is just a late addition. Like a remodeler who shows up 5 years later to add a sunroom.
That is precisely what the data suggests.
Wow.
And the reason the lung cancer only needs a remodeler is because something else already built the house. The study found that tumors harboring these late non-canonical drivers often occur in patients who already have a high penetrance germline mutation.
Let's define that. Germline means inherited from your parents, present in every cell of your body.
Right.
High penetrance means it has a very high likelihood of actually causing the disease. So we are talking about something like an inherited BRCA1 or BRCA2 variant.
Exactly. Because that inherited germline mutation jumpstarts the cancer from the very beginning, the tumor doesn't need to acquire as many traditional somatic drivers to get going. The foundation is already laid.
Oh, that makes sense.
Yeah, this evolutionary freedom pushes other mutations, like our PIK3CA remodeler, into late non-canonical roles just to help the tumor adapt later on.
Think about how radically that shifts a clinical approach.
Oh, it changes everything.
Right. If you are an oncologist looking at a patient's genetic report, you can't just see PIK3CA mutated and treat it the same way every time. You have to ask, am I targeting the architect who built this cancer, or am I wasting my time targeting the remodeler who just showed up?
Exactly. The timing changes everything, and that timing doesn't just dictate the tumor's evolution, it heavily influences the patient's physical timeline as well.
Meaning?
The researchers correlated these specific genetic alterations with the age of disease onset in the patients.
And they found a fascinating divergence between different types of mutations. Specifically, they looked at fusions versus point mutations.
Right. Let's explain those.
Sure. A fusion is when a massive structural error occurs in the DNA, and two entirely different genes literally break and fuse together into a new Frankenstein gene.
Yep. While a point mutation is just a single letter swap in the genetic code. Much smaller scale.
Okay.
And what they discovered is that tumors driven by fusions are associated with a significantly earlier age of diagnosis compared to tumors driven by standard point mutations.
Mechanistically, that makes perfect sense. A point mutation is a subtle typo, right? It might take decades for the cell's environment to align perfectly for that typo to cause a catastrophe.
Exactly.
But a fusion gene is like taking the front half of a Ferrari and welding it to the back half of a tank.
Yeah.
It is an immediate catastrophic structural change that drives aggressive early onset disease without needing any help.
Without needing any help is the key phrase there, because point mutations often do need help.
How so?
Well, the researchers explored this by breaking down the famous BRAF gene into different functional classes.
Okay.
Class 1 is the famous V600E mutation, which operates entirely differently than Class 2 or 3 mutations.
Let's use lung adenocarcinoma as our test case here.
Good choice.
The BRAF V600E mutation is incredibly rare in the lung, appearing in only about 1% of cases.
Right.
But the researchers found that when it does happen in the lung, almost half the time, 47% to be exact, it's accompanied by a co-mutation in a totally different gene called SETD2.
This is the concept of genetic synergy. In a canonical tissue like melanoma, BRAF is a master key. It can force the cell to become cancerous all by itself.
Okay.
But in a non-canonical tissue like the lung, the cellular environment is hostile to it. The BRAF mutation alone isn't enough. It requires an accomplice, SETD2, to help dismantle the lung cell's specific defenses.
So it's a bank heist.
I love that. Yes.
A fusion gene is a master key. It blows the vault doors wide open all by itself, leading to early onset disease.
Right.
But a point mutation like BRAF often finds itself staring at a vault. It can't crack alone. It needs a specific accomplice, a co-mutator, to bypass the tissue-specific alarms. If the accomplice doesn't show up, the heist fails, and the cancer doesn't grow.
It really reveals a staggering level of complexity. We aren't just mapping gene A to cancer B anymore.
No.
We are mapping gene A, requiring the presence of gene C to cause cancer B, but only if it happens in organ D.
So recap time for you listening. Bullet 1.
Yeah.
Canonical drivers are early and foundational, while non-canonical drivers are late evolutionary additions, often popping up when inherited mutations are already present.
Yeah.
Bullet 2. Gene fusions trigger cancer at a younger age than alterations like point mutations, because those point mutations often require strict cancer-specific co-mutations to even function.
Exactly.
So think about that complexity. How might knowing the timing of a mutation or the accomplices it needs change how a doctor chooses to treat it?
It changes the entire paradigm.
Which brings us to the final, and frankly the most profound layer of this deep dive, because the tumor isn't just navigating its tissue environment and finding genetic accomplices.
No, it has an even bigger enemy.
Right. It has to survive the human body's ultimate hostile defense system, the immune system.
And to understand this interaction, we really have to look at the bleeding edge of oncology, which is T-cell receptor therapies, or TCR therapies.
These are actively in clinical trials right now.
They are, and they work by targeting something called a neoantigen.
A neoantigen is essentially a distress signal, right?
Basically, yeah.
When a cancer cell mutates, it sometimes accidentally produces a weird mutant protein fragment. The cell naturally displays this mutant fragment on its outer surface, almost like waving a microscopic flag.
Right.
And the immune system's T cells patrol the body looking for these specific mutant flags. When they see one, they destroy the cell. So TCR therapies are designed to artificially train the immune system to spot these specific flags.
Exactly. But the researchers highlight a massive biological bottleneck here. The cancer cell cannot physically display that mutant flag unless the patient possesses a very specific inherited molecule called an HLA allele.
Human leukocyte antigen.
Yes.
If the neoantigen is the mutant flag, the HLA allele is the physical flagpole bolted to the outside of the cell.
That's a perfect image.
Without the correct genetically matched flagpole, the flag stays trapped inside the cell. The tumor remains completely invisible, and the immune system just walks right past it.
And this is where cancer biology intersects with global health equity in a very stark way. Your repertoire of HLA alleles, your collection of flagpoles, is inherited.
Right.
It is entirely dictated by your genetic ancestry and human evolutionary history.
Because over millennia, different populations were exposed to different regional pathogens, right?
Exactly. And that shaped their immune systems to carry different HLA alleles.
So the researchers looked at specific, highly targeted cancer mutations, like KRAS, G12D, and G12V.
Some of the most notorious drivers.
Yeah. And they mapped out which specific HLA flagpoles are required to display them for these new TCR therapies. And the disparities they found based on genetic ancestry are jarring.
They really are. They found that East Asian patients had the highest frequencies of the necessary HLA alleles for certain trials. But when you look at the KRAS G12V mutation, the data shows that 0% of patients with African ancestry possess the required HLA allele for the current clinical trials.
0%.
Zero. This is compared to roughly 26% of patients from other genetic ancestries.
Just let that sink in for a second. Your inherited genetic ancestry, the evolutionary history of your immune system, literally dictates whether you possess the cellular hardware to even be eligible for these cutting-edge therapies.
It means we cannot design immune therapies based solely on the tumor's mutation. If we do, we risk developing incredible treatments that are biologically useless to entire demographics of the human population.
But say you were lucky enough to have the right flagpole. You have the right HLA allele, the flag is waving, and the immune therapy starts working. The tumor is being attacked. What does the cancer do?
It evolves. The study details a resistance mechanism called somatic HLA-LOH.
We discussed loss of heterozygosity earlier, deleting a gene.
Right. In this scenario, the tumor realizes that its specific HLA flagpole is getting it killed. So it undergoes a targeted mutation to literally delete its own HLA gene.
It chops down its own flagpole.
Exactly. By destroying the display mechanism, the tumor pulls the mutant flag back inside, instantly becoming invisible to the immune system again.
That is wild.
And the researchers noted this happens incredibly frequently in canonical cancers, where the driver mutation is highly immunogenic.
Meaning it naturally triggers a strong immune response.
Right. The selective pressure on the tumor to hide is immense.
It is a terrifying, brilliant game of evolutionary cat and mouse. First, your ancestry dictates if the therapy can even work.
Right.
And second, the tumor can surgically edit its own immune hardware to render the therapy obsolete.
It completely shatters the illusion that cancer is a static target. It is a highly dynamic, reactive ecosystem.
So, final recap. Eligibility for these new immune therapies is heavily dependent on HLA genotypes, which vary drastically by ancestry. And tumors can develop intrinsic resistance by deleting their own HLA alleles to hide from the immune system.
Spot on.
Which brings us to the ultimate takeaway of the MSK-50K cohort. This paper proves definitively that precision oncology has moved far beyond just pointing at a genetic typo and prescribing a drug.
Absolutely. To treat a patient today, we have to understand the tissue context. Is this mutation canonical or non-canonical? We have to understand the clonality. Are we targeting the foundational architect or a late-stage remodeler?
Right.
We have to map out the co-mutations to see what accomplices the tumor is relying on. And finally, we have to sequence the patient's inherited immune genetics to know if our therapies can even interface with the cell.
It takes our understanding from a flat two-dimensional map to a four-dimensional high-definition view of biology that includes time, environment, and ancestry.
Beautifully said.
Thank you so much for taking this deep dive with us today. But before you step away, I want to leave you with one final thought to mull over based on what we've learned.
I love these.
We now know that a mutation's behavior is strictly governed by the specific tissue it starts in. But think about metastasis. When a tumor becomes aggressive and physically migrates from the lung to the brain or from the breast to the bone, well, it is violently invading a completely new tissue context.
Oh, wow.
Do the rules of these canonical and non-canonical drivers suddenly change all over again in the new organ? Does the remodeler suddenly find itself acting as the architect of a new tumor?
It's a profound mystery.
And exactly the kind of question driving the next decade of cancer research.